Distinguished Professor Angela Belcher, ladies, gentlemen, welcome to the 47th Hassel Lecture. On behalf of, I would say, University of Oslo, Norwegian Chemical Society, I have the pleasure to wishing you welcome to this lecture. It's number 47th in the series, and by chance, the element number 47 is silver, the element with the highest conductivity of electrons and heat. So now we know that as well. So the lecture will be given by Professor Angela Belcher at Massachusetts Institute of Technology, perhaps better known as MIT, where she is the Director of Biomolecular Materials. But before introducing Angela Belcher, I would like to give a short biography of Odd Hassel. So the Nobel laureate Professor Odd Hassel, the lecture series here was inaugurated in 1967 on occasion of Hassel's 70th birthday. That was 2 years before he received the Nobel Prize. Keep that in mind. He was born on May 17th in 1897, 120 years old. His bust is up there, in case you know. He studied in Oslo and got his Cand. Real degree in chemistry in 1920, went to Berlin to the Kaiser Wilhelm Institute in Dahlem to conduct X-ray crystallography. He obtained doctoral degree from Berlin, Oslo to work as Universitetsstipendiat. At July 25, 1 year later he became docent of physical chemistry, and in 1934 he became Norway's first professor in physical chemistry here, where he stayed for the rest of his career. Hassel published a lot, numerous papers. During the first year here in Oslo, he dealt with problems in inorganic chemistry. From 1930 onwards, his works was concentrated with problems connected to molecular structures. In particular, his pioneering work on the conformation of cyclohexane and its derivatives. His first groundbreaking paper in this area was published in 1943, and the final crystallographic refinement was ready in 1947. And later, he made numerous contributions to the structure of charge transfer complexes. He was awarded the Nobel Prize in 1969 for his work on the structures of conformation of organic molecules, where he shared the prize with Sir Derek Barton, Professor of Organic Chemistry at Imperial College London. Professor Hassel passed away on May 11, 1981, and it is in this memory that this lecture is continued. The selected Hassel lecture was initially chosen among the structural chemistry, but has since become more or less connected to Hassel's activities. This is in accordance with the chemistry's constant evolution and the fact that many and more interesting developments occur in interdisciplinary areas, which we will hear about today. And now a few words about the present Hassel lecture. Angela Belcher grew up in San Antonio, Texas, where she attended the University of California, earned her PhD in chemistry in 1997. Topic of her PhD thesis was related to the growth and structure of the abalone shells and its pearls, and has since become a token of your identity. After studying the shells, she and colleagues at MIT moved further with engineering the M13 bacteriophage, the virus, to become the workhorse of the future to make our new inorganic materials. That we will learn more of today. Dr. Belcher was awarded the 24th Annual MacArthur Foundation Fellowship, the 2004 4-Star General Recognition Award, 2006, she was named Scientific American's Research Leader of the Year. Her work has been published in many prestigious scientific journals, including Science, Nature, and has been reported in numerous popular science events as well, and popular press, including Fortune, Forbes, Discover, the New York Times, and the Wall Street Journal. She has been recognized as one of the top 100 innovators in the world under the age of 35, and the same under the age of 40. And I think we continue. She's a founder of 4 companies and a lot of interesting research. So, Professor Belcher, without wasting more time, I give you the floor.
Thank you very much for that kind introduction, and it's a huge honor to be here today on this special lecture. I brought some abalone shells with me. So I've been inspired, well, actually since I was in middle school with the evolution, the origin of life, and the interface between organic and inorganic structures. And as was mentioned, I did my PhD on how abalones grow shells. Abalone are biocomposite materials. They're 98% by mass calcium carbonate, 2% by mass protein, yet they're 3,000 times tougher than their geologic counterpart. And they're made in ocean temperatures and ocean pressures. They don't use toxic materials, and they don't add toxic materials back into the environment. And so in my group, we think this is a fantastic model for how to manufacture and think about how to make new kinds of materials. I'll talk a little bit about abalone shell today and how we got our inspiration from abalone shell and how we're using that inspiration to make other kinds of materials we like to say nature hasn't had the opportunity to work with yet. Let's see if I can. Oh, okay. So in my group, our goal is trying to give non-living structures more lifelike qualities so they can enjoy some of the same qualities that we do in life in terms of being able to make copies of themselves, being able to pass on genetic information, but also the idea of environmentally friendly synthesis of materials. And so I say that this is my dream team of students. I've been really fortunate over the last 10, 15 years to have the most wonderful group of graduate students and undergraduate students and postdocs to work with. And so I'm a professor, they really do all the work, so the work that I'm going to talk about is the work they did today. I'm also in biological engineering, material science engineering, and the Cancer Institute. So what we do is we call genetic control of materials. And going back and thinking about that abalone, when a male and female abalone get together, they pass on the genetic information that says this is how to build an exquisite structure. Now there's 2 secrets to that abalone shell, actually really 3 secrets to that abalone shell that make it so fantastic. One is that it's a nanostructured material, and I'll show you some of what that nanostructure looks like in scanning electron micrograph. It's also had about 50 million years to get good at making this material through trial and error. And it's that 2% by protein that really is kind of magic that actually helps control this material. So when a male and female abalone get together, they pass on that genetic information that says this is how to build an exquisite material. Go on and make your own. Same with diatoms that are made out of glass. There's at least 10,000 different types of diatoms. They differ from each other based on their morphology looks like, this star-based diatom or this kind of space-shaped diatom. Every time they reproduce, they make another star-based or kind of a spaceship-looking structure. So they have the DNA sequence that codes for the protein sequence says this is how to make that kind of material. So when we think about future materials, we start at the beginning of Earth about 4.5 billion years ago. It only took about 1 billion years to have life on Earth, but then really amazing things started happening. You started having multicellularity, you started having photosynthesis, and organisms were doing really amazing things. But it wasn't until this geologic time period called the Cambrian geologic time period, the explosion of life, that organisms in the ocean started making hard structures. For these billions of years before, everything was soft and squishy. So why is that? Why did it change? Well, it was during this time that the ocean started changing. There was increased calcium, silicon, iron in the environment, and the organisms started using it to make materials. And so what we've been interested in my lab is how do you give genetic information to solar cells or batteries, for example, so that they can also learn how to genetically program or template those materials. And so a lot of times I have a picture of one of my kids here. I have two kids, and I say they're incredibly complex organisms. It's hard to get them to put their shoes on in the morning. So if you want to think about how to genetically engineer an organism to work with a semiconductor material or a battery material, let's start with very simple organisms. Let's start with bacteria, and let's start with viruses. And so this is a cartoon of the kind of virus that we work with. It's called M13 bacteriophage. What if you had a virus that had the genetic code to become a battery electrode, to wire itself, and be able to make copies of itself through its bacterial host? And so in my group, we focus on the interface between nanotechnology and biomaterials, and we're very application-oriented. So we're interested in problems that we think are the most pressing problems on the planet. We work on energy and healthcare and the environment. And for years, people have said I should focus. People still say I should focus, but I feel very focused. I feel like I focus on trying to use biology to make better nanomaterials, where those nanomaterials have improved performance. And so people here know a lot about nanoscience and nanomaterials, but some people think, wow, nano is great because it's small. But the reason nano is great is because when you take a bulk number of atoms and you start to get to where you have collections of smaller numbers of atoms, the way those atoms interact is different. It changes the optical properties, the electronic properties, the mechanical properties, the magnetic properties. And so we're interested in the nanoscale. We're interested in biology, controlling materials on the nanoscale so we can improve device performance. And so we work on solar cells and batteries. We work on drop-in fuel replacements, enhanced oil recovery, CO2 sequestration storage, more abundant materials, cleaner mining, water purification, therapeutics, and imaging. And that's where people say you should focus on just a few things. But to us, most of the problems in the world have material solutions. And so if we can find the right materials and control those materials in a way that we can have an impact in all of these areas. And so these are some companies I've founded or co-founded: Siluria Technologies and Cambrian Technologies, and two new companies coming out of my lab this year, Geyser Biotech and Nirvana Diagnostics, all based on using biology to control materials. And so the kind of nanomaterials I'm going to talk about are DNA, which is the genetic code for the synthesis of our materials, single-walled carbon nanotubes, and different kinds of nanomaterials that we're going to make, and I'll talk about them along the way. But I've been interested in using biology for this kind of control and synthesis because biology already exists on the nanoscale. Inside of our cells, you have different types of proteins, and you have antibodies and DNA. They're already at the absolutely perfect size scale for controlling materials. And so what we've been working on doing is just harnessing them to give them new ability to control whatever we're interested in so that we can work on assembling devices and solving problems. Now, going back to that abalone shell that I passed around, if you took this red abalone and fractured it, what you see is it's made out of these stack of coin-like structures. And this is aragonite. It's a metastable crystal structure of calcium carbonate. And they stacked on top to make this beautiful brick wall structure. So you see where the toughness of this comes from. Now, the thickness of these tablets is a function of the particular species of abalone. And other abalones can be thinner, or other ones can be thicker. And it's where you also get that beautiful colored property from the abalone shell. Now, between every individual tablet of calcium carbonate is a protein. This protein has several functions, and there's several different proteins, but they're involved in nucleation of this material and control of the synthesis and have a dampening effect that's part of the toughnesses as well. And I talked about diatoms. Other examples of natural biomaterials are iron oxide, Fe3O4, Fe2O3, used in navigation. And if you look at different views of this through scanning electron microscopy, you'll see these individual tablets. You'll see proteins that are actually in between the tablets that actually help set the spacing, as well as proteins that where nucleation goes through these individual tablets to the next tablet. And this is actually what it looks like in the top of a pearl. So this is organic protein with the smallest calcium carbonate that's starting to grow out before it reaches its full length. When I was a young professor, I worked in the black pearl industry. It was actually paid a lot better than being a professor. It's how I bought my first house. But in the black pearl industry, they want really high quality pearls. And one of my jobs was to look at different ways the animals were treated to understand what gave the highest quality $3,000 pearls as opposed to your $50 pearl or such. And it was really, really interesting because it was really a function of how much organic there was in between the individual tablets and when mistakes happened and how quickly the organism could recover from the mistakes. And I would take like a several $1,000 pearl and I would cut it in half and image it and look at it in an electron microscope, collect its proteins to see why one was a very high quality pearl and one wasn't. But the organisms in the ocean, they only had a limited number of elements to work with, and they had to work with what they had. This is from a NOVA special that I was on, where we were talking about the elements that came from the ocean, and wouldn't it be great if organisms could use much more? So organisms in the ocean use calcium in the form of calcium carbonate and calcium phosphate, use silicon in the form of silica, iron oxide, Fe2O3, Fe3O4, but didn't really use these, of course, as enzymes, but didn't really use these as materials, and didn't use materials over here or down here. And so what I wanted to do was give organisms the opportunity to work with most of the periodic table so that we could work on genetically coding them and using them to form different materials. So I had passed out some periodic tables because some people already know this joke, I guess, from a TED talk that I gave. But I was really lucky that President Obama came to visit my lab, and he's a really smart guy. He asked a lot of great questions. And when I wanted to give him the periodic table that I handed out, it says, welcome to MIT, now in your element. Because when I first got to MIT, I finally felt like I really fit in. And so I had them made for the freshman incoming class every year. And on the other side are the amino acids with their pKa's. And so I thought every freshman should have a periodic table and the amino acids with the pKa's. And so when President Obama, once he finished, he was visiting the battery work in my lab and the solar cell work in my lab, and I wanted to give him the periodic table. And I was actually kind of nervous. I'd stayed up the night before thinking about how you bring up the periodic table in conversation with the president. And so I said, sir, I want to give you the periodic table in case you're ever in a bind and need to calculate molecular weight. And he said, thank you, I'll look at it periodically. And he went off to give a lecture later that day on clean energy, and he held it up. And so I wanted to give you the same one so you have the presidential periodic table. Some people said, oh, there's elements since then, but I stopped adding elements once I gave it to President Obama. So I talked about the abalone and how it had this amazing ability to grow materials based on the fact that they had protein. So these proteins are made out of those 20 amino acids that are on the back of your periodic table. And the way it works is that organisms in the ocean may have this organic matrix, this protein. In the case of the abalone, the one that actually grows this calcium carbonate-based material, are very negative charged amino acids. They have rows of aspartic acid or glutamic acid, and they can grab that calcium from the environment. They can put down calcium carbonate, and they can help with the nucleation of these materials, but they can also act as poisons to tie up certain crystallographic growth. And so if anyone collects calcium carbonate isostructures, okay, so I'm the only one. So basically, if you've seen geologic aragonite, it grows as this pillar, this hexagonal pillar. But when I showed you how it grew as an abalone, it grew these thin tablets. It's because it grows these thin tablets that are laterally offset, and it gives us this really tough structure. So the proteins can actually control not only which crystallographic structure is grown, but also what orientation is grown, and make certain axes longer and certain axes shorter. And so what I wanted to do was I wanted to be able to have a protein that could control any structure I wanted to. And I wanted to think about how to do that. And so I turned to a well-known biological technique called Phage Display. So Phage Display uses a virus called M13 bacteriophage. It's a virus that only infects bacteria. You have it on you. You've eaten it today. And the reason I like it, I'm really a material scientist, so it's an object that's 880 nanometers in this direction and 6 nanometers in this direction, and it has single-stranded DNA. The single-stranded DNA codes for the proteins that make it up. And so you can go in and make a modification in gene 3, and that modification in gene 3 will make a modification in protein 3. So you can put a random DNA sequence in here that codes for a random peptide sequence. And so if you do that a billion times, you have a library, a way for looking for proteins that affect inorganic structure. So again, looking at M13 bacteriophage, we have these different genes. These different genes code for these different proteins. And so we can make modifications in this protein, modification in this protein, or modifications in this protein. So you can make a library. This is a gene 8 library, which is the major coat, where we can take random DNA sequences and splice them in and have a virus that will make millions of copies of a protein with a modification in its coat. And so what this allows us to do is to take about a billion viruses and a couple of microliter sample and force them to interact with anything we want. Semiconductors, insulators, metals, magnetic materials, conducting polymers, carbon nanotubes. What we're looking for is a virus that expresses a protein that recognizes a semiconductor material or a battery electrode based on arrangement of the atoms and based on size and charge. Now, most of them out of that billion will not interact. We wash those away. Anything that does bind, we remove from the surface either by ion competition or lowering the pH. Now, viruses can't make a copy of themselves. We infect it into a bacterial host, make a million copies, and then we isolate them. And now we have a population of viruses that recognize on their tip this particular material. And now we ask them to bind again. And we go through this kind of Darwinian process, looking for the 1 out of a billion that will grow a better material. And so when I first came up with this idea, I was a young professor sitting in my office, and I submitted it for a grant to the United States Army. And we got 2 reviews back. The first one said, "She's insane," with an exclamation point. And the second one says, "It's an interesting idea, but I don't think she has the background to do it." And I was really lucky that the Army funded me anyway. And we developed this technology in my lab. And then eventually, I won a presidential award for this, and went to the White House. So it was called crazy. I was very crushed by it at the time, but we went on to develop technology around it. So I'm telling you that in part for the young people in the audience, just starting out, that I think it's okay to be crazy. And so this is a cartoon that my students made that just kind of show you take a whole library of information of phage and you throw them at a material. You're looking for ones that are going to recognize the surface. Most of them won't recognize the surface. Those get washed away. You need to remove these from the surface by disrupting the protein interaction at the surface. And now you infect them into a bacteria. The bacteria acts as it has the machinery to make lots of copies, acts as a factory to make lots of copies of that particular virus. Now that virus you can use to bind to materials or try to grow materials of interest. Now if you go and look at what the code of that virus looks like, it's made out of these beautiful alpha helixes that surround the DNA. And these beautiful alpha helixes are actually crystallographically related to each other around that DNA sequence. And so what we were doing is going in and making modifications of proteins on the surface with a billion different possibilities to look for one that can grow a material of interest, just like the abalone was growing that shell. And so looking in again, kind of pulling away some of those alpha helixes, you can see the kinds of functional groups that you can have within these alpha helixes. And so this is the reaction vessel where the nucleation, the growth of material is going to happen. This is one of my best collaborators, Paula Hammond, and this is a cartoon of what my students I think we look like, and I'm always wearing the same thing. I'm wearing the same thing I was in this picture. And so we're trying to give genetic information to solar cells and batteries, to catalysts, drop-in replacement fuels, CO2 sequestration storage. And one of the main areas of my work right now is for cancer diagnostics and treatments. And we also work in artificial photosynthesis as well. Again, going back and looking at these M13 bacteriophage, they look like wires to me. They're actually beautiful nanowires, and you can use them as nucleation points to grow materials. And so in this case, it just shows a diagram of the idea of being able to take ions from solution and nucleate nanoparticles with controlled size. This is one particular sequence that we isolated from my lab, that once you isolate this, you can add gold ions from solution with this particular sequence called P8 number 9. Here's the sequence for it, and it's very good at growing gold nanowires. Actually, 98% conversion of these ions in solution to gold nanowires. You can control their diameter. This is transmission electron microscopy image. And then you can go in and start doing other fun things. You can take different sequences and different kinds of ions and make different alloys of these materials, as well as core shell structures, based on controlling both the amino acid sequence from your virus, as well as the chemistry that goes in. In this particular example, we're growing a gold nanowire, and we're putting platinum nanoparticles on top, and we use this for fuel cell applications. This, I think, is just a higher resolution image showing the platinum nanoparticles on the outside. And so we wanted to test this for EOR and OR reactions, and so we could have we tested different richnesses of gold versus platinum in different combinations for these fuel cell applications. And so this is looking at some of the fuel cell activities, and this is for oxygen reduction reaction here, and this is benchmarking against platinum. And as we vary the DNA sequence, and as we vary the concentration of ions, we can greatly enhance the activity, but it's a different activity needed for the ethanol oxidation reaction. Here we have a gold that's 2.6 to 1, showed us the best example in this case. And so this is just showing you that we can take these DNA sequences, build viruses out of them that have protein sequences in them that can attract atoms, that can grow nanoparticles in different combinations that can be used in applications. And this is some of the list of the kinds of materials that we use in our lab that are not the kind that you would normally think of as biological materials. We've used many of these in energy storage and catalysis and solar applications and magnetic materials as well. But one of the things that we like about the M13 bacteriophage best is that it is this squishy structure. So you can think about actually having it do multiple things at one time. You can think about it having grow or bind a nanowire, and then simultaneously grow a nanoparticle. And so you can think about if you have, say, carbon nanotubes in this case and titanium nanoparticles, you can think about mixing them at the nanoscale. You can think about physical mixing or chemical modifications to the surface. But in this case, you may not get very homologous mixing. In this case, you might actually affect the surface in a deleterious way. And so what we use this virus as a mediator to have one gene that picks up the carbon nanotube, another gene that grows titanium nanoparticles. And so we spent the last several years in my lab thinking about how do you go from a DNA sequence to a functioning device. And this particular one is used for a dye-sensitized solar cell. One of the first applications we worked in was in energy storage for lithium-ion batteries. We worked on making anode materials and cathode materials for this. It's still a major focus in my group today. We work in many different kinds of batteries, from very small batteries for very small UAVs. A lot of the work in our lab is based on unmanned vehicles of this size for structural batteries. We work on wearable electronics, and then we work as well in batteries that can be used for lithium-oxygen batteries. My students want to go to grid and microgrid. We're working on sulfur batteries and other kinds of batteries, but I'm not as optimistic about getting to that large of a scale. To make the first kind of virus batteries, what we did was take a virus, and we selected for the ability to grow cobalt oxide nanoparticles. These nanowires will naturally self-assemble based on their anisotropic shape. And then we actually go in and we do additional engineering. So if you start with the ability to grow a material of interest, in this case it's cobalt oxide, if you're able to grow it but you want to improve it, you can either go through additional genetic engineering or you can do protein engineering to add other kinds of groups. And so then we wanted to actually have it assemble into a battery. And so this is all done in a shaker using kind of traditional biological tools. In this case, it's a single virus-based nanowire with very small crystalline cobalt oxide that's all nucleated on top of it. We used that material on copper with a separator and electrolyte, and a lithium as a counter electrode. And this is a commercially available nanomaterial. This is a theoretical capacity for 8 lithiums per cobalt oxide. And you can see that we hit theoretical capacity, but as we started to cycle, it started to fade. This is looking at what it looked like grown biologically, and this is what it looked like made out of a powder under the same conditions, room temperature, room pressure, kind of synthesis. And so it wasn't great, but we had a reversible capacity of about 600 to 750 milliamp hours per gram on our first go at it. My students decided to go on and give it a second gene to grow a second kind of material. They took a protein that we had shown that was good at binding gold, and they put it in a low copy number into the virus, so now it had the ability to grow two different materials simultaneously. So this is our first generation battery, and this is our second generation battery, so it improved in terms of capacity, in terms of faster reaction rate. We were pretty happy, and we went to present that as the first virus-based battery to the Army, and they said they weren't impressed, and they said, you only made half, where's the other half? So I went to my students, because I don't do any of this myself, my students do all the work, and I said, where's the other half of the battery? So let's start working on cathode material. So this is my two students, Yoongjung and Hyungjung, who decided what we really need to do is take M13 bacteriophage and get it to pick up single-walled carbon nanotubes to be good electrical conductor, and then get it to grow iron phosphate-based material. And so I said that sounded like a good plan. They were able to engineer viruses that could pick up single-walled carbon nanotubes on their tips and then simultaneously grow iron phosphate. And these actually worked as the electrons were electrons from the current collector were able to be transported in this kind of super highway, and we got much greater improvement with this kind of material. And then here's specific energy versus specific power. So this is our 1-gene material, this is our 2-gene material, and this was our best 2-gene material at the time. And so I was pretty happy about this, and we were publishing it in the journal Science, and it was accepted with revisions, and I got a phone call that said, your battery is going to go to the White House. And I said, okay, well, let me talk to my editor. So I called my editor of Science, and I said, is it okay if the president of the United States, I said, is it okay if the president breaks the embargo? And he said, the president of MIT? I said, no, the president of the United States. And he said, no one breaks the embargo for science. And he said, if you can get the revisions that you're supposed to get done, and then we'll post it when it's done. But meanwhile, I told my students that, we need to send your batteries to the White House. And so they said, we actually use them all for the paper. We don't have any good batteries. And so they had to make them. They made them overnight, and they FedExed them off to the White House. But then we didn't get invited, just the batteries got invited. And then they said, actually forgot to test them. We didn't have time to test them. We just shipped them. And so I was pretty nervous. We had a special battery holder made and everything. And so we were watching on the internet because we weren't invited. We had to watch it on the internet. And then it actually worked, and we were really, really happy about that. But then we actually started thinking about, well, it worked with carbon nanotubes. What about graphene? We took some sequences that we already had, did some specific mutations in them, and were able to get viruses that bound to graphene and then simultaneously could grow other materials. And so this material was bismuth oxyfluoride-based material. And we found only when you had the virus in contact with the graphene plus growing the material, you had the best kinds of data. So now you're looking at specific capacity. And this is actually looking at the virus with growing the bismuth oxyfluoride by itself. This is actually looking at just precipitated bismuth oxyfluoride with graphene. This is growing the two together, and then this is actually growing them separately and then putting them together. And it was when they were in the closest contact that we had the best kind of data. My students went on and spent a lot of time building foams out of these. This is a copper-based foam and a nickel-based foam that are freestanding foams of these different kinds of materials. And then once we have these foams, making hydrogels, from hydrogels making foams, and then starting to coat them with other materials. So in this case, you have nickel nanofoams. On the outside, you have lithium manganese oxide based materials. So those materials we're using right now for what we are calling structural batteries, where the battery of where the wing of the UAV is the battery, and we're working to try to increase the flight time of this particular UAV that is currently used in in Afghanistan, and we're trying to double its flying time. So we had a colonel in the Army come into my lab saying that these are actually very, they don't last long enough, and they're very noisy. Can we work on trying to use biology to improve this? My students also said, we'd worked on lithium-ion batteries. They said we really need to work on lithium-oxygen batteries or lithium-air batteries, with the idea of here's the current state for lithium-ion. Here's where lithium-air could be. And so we said, well, we really need to work on these kinds of materials. But there's a lot of limitations to these. There's large overpotential, low current density, cycle life, and cost efficiency. So what my students decided to do was to work on the catalyst electrode for a lithium-oxygen battery electrode, and see if we could grow one that would perform well. And the idea was that it would also be good at allowing the reversibility of the lithium peroxide during this reaction. And so here's what our virus-based manganese oxide nanowires look like. They actually have this very spiky structure. This is actually just precipitating these materials, and this is grown biologically. You can see the surface area is greatly increased by this. This is looking at some initial data for our biotemplated versus our deposited manganese oxide-based material. All of these, of course, are made at room temperature. Then what we decided to do was to add a little bit of another element, either gold or palladium, to these structures. You can either do it through biological means, or you can do it through adding a little bit of a polymer that can attract these nanoparticles. This is a, this is a cartoon my students made of what the idea is that you have a virus that's in water, it's in solution. You have modification of each of these proteins that allows for the nucleation of these manganese oxide materials. You have manganese, you grow these spiky kinds of structures. You add in a little bit of palladium to this, and then the reaction you have is lithium plus oxygen plus 2 electrons, forming the lithium peroxide. That's reversible. So that's a single wire. Now you make billions of these wires. And then we can test it as a lithium-oxygen battery electrode. And that's my car. No, this is a car my kids wish that we had, but I actually have a Chevy Volt, which I love. But the idea is that you'd be able to go twice as far. And so what we actually saw from these experiments, we had 45% improvement in the specific capacity, and we had 7 times higher current density than previous manganese oxide electrodes. And it worked best when we had the combination of the biotemplating and the addition of the high surface area and the addition of the palladium nanoparticles. My students have then gone on to try to make aerogel-based materials out of viruses, and this is a lithium-oxygen battery electrode that's an aerogel, and you can actually compress it to 50% its size, and then it recovers, and it still has the same performance. And it's all based on the ideas of being able to make hydrogels out of these materials, and then from the hydrogels be able to make aerogels. We also worked in dye-sensitized solar cells with Paula Hammond. And my students came to me and said, okay, well, I think that we can make a difference in dye-sensitized solar cells. In dye-sensitized solar cells, you have titania particles. You have a dye, you shine light, you promote an electron. The electron gets injected into the titania, and it has to make its way through this titania nanoparticles to the current collector. And it has a electrolyte that's for recycling. And so what my students said was, let's see if we can help this electron find its path. So right now you shine light and the electron makes its path, but it can get recombined with the electrolyte, or it can get recombined with the dye. So in a structure like this, you have high surface area, but you have high dye loading, but you have random electron path and a short diffusion length. So the idea was to add carbon nanotubes to these kinds of structures, and people had worked on this for years and found that the carbon nanotubes had actually decreased the efficiency, not increased the efficiency. So what we worked on was using semiconducting carbon nanotubes and using a virus to help assemble these materials in the right kinds of locations. And so the idea was that you would have the electron that would find the virus-based carbon nanotube complex and then be able to find an easier path. And so in order to do this, my students took M13 bacteriophage, and they engineered the carbon nanotubes to sit in all these pockets along the outer surface of this material. And it ends up that the virus actually acts as a surfactant to be able to keep these nice and stable in solution and still have spaces to grow other materials like titanium. So they started with a library of M13 bacteriophage, and they looked for ones that could keep carbon nanotubes in solution to act as a surfactant. And so in this case, this is basically dialysis tubing. These are different clones that they pulled out that looked like they bound to carbon nanotubes, but it was only these that actually kept them soluble. And then we looked at the different sequences of these and made decisions in terms of which ones would be best for binding carbon nanotubes and growing the titanium nanoparticles. And it acts like a surfactant, and so it was actually the particular choice that we made. And so now what we have is this is a transmission electron micrograph with a virus picking up a single carbon nanotube right there, and then we will grow titania around this structure. And based on doing this, this is with no single-walled carbon nanotube. This is with the M13 single-walled carbon nanotube, just a surfactant, and as produced. And based on this, we're able to get increase in 33% efficiency of the dye-sensitized solar cells made in our group. And it was 0.1% biology that led to the increase in this solar cell efficiency. Another area that people have asked me to work on for many years was biofuels. I'm not a real biologist, and so I was worried about keeping things alive. And so I said, well, what if we use biology to make new kinds of inorganic materials, and then from there, what if we go directly into fuels? And this was a company that I'm the academic founder of called Siluria Technologies. It's a company that developed a low-temperature oxidative coupling of methane to go from methane into ethylene, ethylene into plastic or drop-in replacement fuels. We started off by working with biological templates and selecting the ability to grow and bind to catalysts, and then high-throughput screening. We're now in 4 locations. We have 3 locations in California, and we have a demonstration plant in Texas that uses air or enriched air or pure oxygen plus methane to form ethylene, and then ethylene we go into gasoline or diesel. And this is the opening of our demonstration plant in Texas that's making a ton of ethylene a day. Another area that I've been interested in for many, many years was environmental remediation. And this, I'm going to talk about this more tomorrow, but we've been working on using yeast. The problem was too big for viruses. We've been using yeast to clean up wastewaters and nuclear spills and disaster spills. And one reason I really like it is we use yeast, the same kind of yeast you use to make beer or bread, which you can store for 10 years or longer. And we're working on increasing the ability of these yeast to uptake atoms or materials of interest and or biomineralize them. And so the idea is for the developing world that you could grow your own water filters that were specific to your particular contamination or your particular well. There's a company that spun off on that. We'd worked on yeast before. We worked on using yeast to make tiles, going from CO2 into building supplies. And so we had worked on genetically engineering yeast to take CO2 and build calcium carbonate tiles, which was being used by Armstrong Flooring. And then about 5 years ago, my boss, the president of MIT, asked me to start working on cancer. And I didn't know anything about cancer, but I said that we would move into the new Cancer Institute and start thinking about cancer. And so we built this group that was materials design and near-IR imaging in cancer, along with another group at MIT called MIT Lincoln Labs and Mass General Hospital. And because I didn't know anything about cancer, we decided to start working on carbon nanotubes and seeing if we could use those in different kinds of imaging applications. And so this is looking at different ways of optically imaging right now. You have MRI, and you have CT, PET, ultrasound, near-IR, which is 650 to 950. We decided to look at second window near-IR, so between 950 and 1650, which has long wavelength, has really good penetration depth, is inexpensive, non-radioactive, and has quick imaging times. The downfall for this was that there was no commercially available equipment for medical imaging, and it required sensitive equipment. And so we decided to start focusing in that area. What we liked about second window near-IR, it's imaging around here, and at this wavelength, it's much more transparent to blood and tissue, and it was suggested that we'd be able to see up to 10 centimeters in depth using pretty inexpensive lasers. And so we decided to develop new contrast agents and build new instruments to be able to look in this area. Now, when you're looking at, there's usually a trade-off between resolution and depth. So you exclude scatter when you're at hundreds of microns. You ignore scatter here. But when you're looking at much deeper depths, you need to embrace scatter. So we started building instruments and probes to be able to image in this particular wavelength. A physician from Mass General Hospital said we should start working on ovarian cancer because there's 250,000 women diagnosed yearly worldwide, and 75% of the patients present with advanced stage, 80% are resistant to chemotherapy, and the vast majority of the patients will relapse and become drug resistant. And there's been minimal increase in survival over the last 30 years. And so we decided to start working with him to see if we could develop technology around this area. Looking at the survival rate in ovarian cancer, the overall survival rate is 46% compared to breast cancer, which is 89%. If you catch it early, you have a 92% 5-year survival rate. But if it starts to spread, it goes way down. The unfortunate part is most of the time, ovarian cancer is diagnosed at late stages. And the gold standard for the contrast-enhanced CT is about 1 centimeter where ovarian tumors are found. And so we said what we want to do is find tumors that are below that size so that we can get it at stage 1 or stage 2. And if we can get them at those stages, we could increase the mortality by 50%. The way that ovarian cancer is diagnosed right now is contrast-enhanced CT. One of the reasons it's diagnosed so late is that it doesn't have a lot of symptoms. The symptoms are very amorphous. There's no yearly screen for ovarian cancer. And the treat for ovarian cancers is called surgical debulking, where the surgeon goes in and opens up the patient and removes everything he can see or he or she can see by eye, follows up with chemotherapy. Now, this is looking at metadata for survival. It's called debulking, based on optimal debulking and suboptimal debulking. So one, you want to be in a really good hospital where they have a lot of experience doing optimal debulking. And two, you want your tumors to be in a place where you can visually see them. Now, before I started working on ovarian cancer, I didn't realize it was a distributed disease. I thought you have an ovary, you have a tumor. But ovarian cancer gets spread all around this place called the intraperitoneal space. And so what we wanted to do was design a technology to help the surgeons find smaller tumors to see if we can increase survival rate. Our answer is usually always starts with the M13 bacteriophage. So we took an M13 bacteriophage the same one we used for solar cells, that was good at binding carbon nanotubes, and we engineered it to bind to a protein called SPARC, which has been shown to be highly overexpressed in ovarian cancer. Now, the thing about single-walled carbon nanotubes and semiconductor single-walled carbon nanotubes is they're fluorescent in this region above 1,000 nanometers, and the chirality of these carbon nanotubes gives a discrete optical signature in that area. So then my students built an imaging system with the indium gallium arsenide detector, an 808 laser, where we could screen for the ability to find ovarian tumors and then look at them in mouse models. Now, this is looking at our first generation imager, and this is three different mice, yes. And this is looking in visible light, this is looking in near IR, and this is looking in near IR2. And so you can start to see that we can pull out features in NIR2 that you couldn't see in NIR1. And these are based on animals that have xenograph-seeded tumors that were allowed to spread. Still in our first generation imaging instrument, this is a single animal, pre-injection, so you see no background fluorescence in this area. Then we inject the animal with our M13 bacteriophage that has the carbon nanotubes, and this is what, where all the tumors are in this intraperitoneal space of the animal. It has a very high burden for ovarian cancer. We don't show this to our surgeon. He just takes the animal and he does surgery, and he closes it back up, and he missed all these tumors. And so then we give him our physical map of where we think the tumors are, and he goes back in and he removes more tumors. He can find more tumors. And then the surgeons we worked with said we really need a real-time surgical guided instrument. So this is a little more data here. And so this is showing that our surgeon couldn't see tumors that were less than 1 millimeter in size, but we could actually pick them up that were less than 1 millimeter in size with our instrument. And so this is unguided versus guided. And so the suggestion was to build a real-time surgical guided instrument, and so we teamed up with MIT Lincoln Labs and another team at MGH and designed the instrument with the surgeons there, still in a mouse model, where we would have different kinds of beams, visible beam, and we still have an indium gallium arsenide detector, and we're able to make it so the surgeon could see, do the surgery in real time and monitor it. And so this is an animal that you're looking at tumors.
I want to see at this point on the monitor we can turn on the in-band source, and that will make it more visible on the screen what he's doing.
Yeah, I think we could do that.
It will be confusing if we do that.
But yeah, the fluorescence will be off, right?
Yeah, it will interfere with, it's like 20 pixels wide what he's actually doing.
I don't actually know how to
It's
turn off
looking-
the
trying to
volume. Okay, thank you. I guess we have to turn it off. Okay, so he opens up the animal. He's able to see it using 3 different light sources, and then is able to remove the tumors. And then you can see that no tumors are left to be seen. And so now we can actually color code these in real time. So we have tumors between 1.5 and 2.5, which we call large. Remember, large is 1 centimeter. We have tumors between 0.5 and 1.5, which we call medium, and we're able to tag tumors that are less than half a millimeter in size. But then we had to actually move on to survival studies. And so what you're going to see is on the top an animal that doesn't have guided surgery, and the bottom an animal that does have guided surgery, both 2 days pre-surgery. So the bright spots are where the tumors are. This is 10 days post-surgery. So this animal, we missed a lot of tumors in the non-guided surgery. It looks like this animal actually has no tumors left, and so we're pretty excited at this stage. Usually in a human patient, the patient at this stage would be followed up with chemotherapy. Our animals didn't get chemotherapy. This is looking 3 weeks post-surgery. This animal has a very high tumor burden, and this animal actually, see these tumors that we missed. So we missed tumors that are less than 0.2 millimeters in size, and they've started to come back. This animal dies 5 weeks post-surgery. It's a very aggressive disease, and it continues to spread. So ultimately, this animal died of ovarian cancer, just like most ovarian patients die of ovarian cancer. So if you look at our survival data from this, we have a 40% increase in survival rate in guided surgery, which is the physicians say is quite substantial. And I have a meeting, I think, next week with the FDA, where we're trying to get this into small-scale clinical trials. But as usual, I say my students do all the work, so I don't do anything, I just direct. So after they built this instrument and they got 40% survival rate, we say, really good job, but I want 80% survival rate. I'm not satisfied with 40% survival rate, so how do you get it farther? And so there's a couple different ways that we've worked on. One is by following up with chemotherapy, and the second way is to get those tumors that are too small to see, that are 0.2 millimeters in size. I can't tell you more about it right now because it's being recorded. But so we've been developing ways of trying to clean up those very, very small tumors with different kinds of nanomaterials that we've made. And I've asked my students to get to 80% in the next 18 months. We'll see where that goes. But we've extended this technology to other kinds of tumors and other kinds of cancer as well. One of the great things about M13 bacteriophage is that it's always the same length. It's 880 nanometers in length. The length of the virus is a function of its DNA. But 880 nanometers in length is actually kind of long. Are there any? There has to be polymer chemists in here. Anyone polymer chemist? I tried to be a polymer chemist for a short period of time, and I wasn't very good at it at all. And one of the things, probably because I wasn't very good, but one of the things that always frustrated me is size selection of the polymers that I needed. And one of the things I do like about biology is biology comes in exact units. Because your enzyme is too short, that's bad. Every time this virus is replicated, it's 880 nanometers in length. And so what my students did was they started making what they're called iniophage, which I think means small in Portuguese, small phage that are between 50 nanometers in length and hundreds of nanometers in length, with the idea of we'd be able to take those and they would circulate longer in the body, and they may be better in diagnostics. But at the same time, we needed short carbon nanotubes, and so I asked my students to figure out how to make short carbon nanotubes. And one of the ways they did was they made DNA origami, DNA folded structures that fold always to the same length. And they put on their DNA origami these other kind of natural DNA structures called G quadruplexes that were linked in really particular locations on the DNA origami. And then they added hemine and streptavidin, and they were going to use this as particular locations in addition of peroxide to break carbon nanotubes. And so they would supposedly only break in certain locations. We're still working on this, but we're able to get enrichment for 65 nanometers in length, which is the length that we're trying to get for our short phage for our cancer imaging. And so this is a DNA origami here that has all the structures together, and a carbon nanotube is along the lengths. And so you add your hydrogen peroxide in, and it cuts at only a certain lengths. We've started using that for pediatric human gliomas, pediatric brain tumors, where we're trying to deliver drugs, image brain tumors, and deliver drugs directly to locations across the blood-brain barrier. Again, I said, good job to my students for designing these, all these instruments and showing that it works. But what I really want was I want one that can actually do depth real time, that's high resolution, high scanning, tissue all the way to the cellular level. I also want inexpensive, minimal invasive diagnostics, and I want to be able to track a single cancer cell in a non-invasive way. And so my students have developed a 5th generation instrument, which we call Dolphin, which I'll talk a little bit about tomorrow, where we can see in depth of 9 centimeters now. Why do we want to do that? Well, the people that we work with at Mass General Hospital, Dr. Beer's group, said that the main way that ovarian cancer is treated right now is through this surgical debulking. And the reason it's treated that way is there's no way of catching it earlier. What we would like to do is take patients, once they've already been surgical debulked, and screen them yearly for reoccurrence. Or we'd like to be able to develop a screen for someone that has a higher percentage of ovarian cancer to develop it as a pre-screen, like a patient would get a mammogram. We want to be able to have an ovarian cancer screen. So that's what we're working on now based on this newest technology. We're not even close, but we can see 9 centimeters in depth. We're not using carbon nanotubes. We're using other kinds of materials, but that's where we want to be. These are my incredible students over different times, and my great collaborators. That's my family in Iceland, our favorite vacation spot. We're climbing a volcano, and this is some of the funding for this project. So I know I covered a lot of material. I thank you for your attention, and I'd be happy to take any questions.
Thank you very much, Angela, for a very nice lecture. And I think there will be some questions. Sorry? Okay, sorry. Yeah. Ute first.
Thank you. Very inspiring talk.
Thank you.
I was thinking of your periodic system. When you choose the elements that you want to use for the batteries, do you have a certain strategy? Do you take what is known to work in from inorganic systems or randomly, or how do you do it?
So we start with, well, when we first started working in batteries, we always collaborated with other battery people at MIT, and they would tell us what the interesting kinds of materials were to work with. And so the materials we used, lithium iron phosphate, lithium manganese oxide are pretty typical materials that are used. Another way that we approach this is we'll have people say these are sets of materials we'd like to be able to make, but they have limitations. And so then we'll try to see if we can select the ability of our viruses to build them in a different way, to make the nanoparticles smaller. Usually when people make batteries, including lithium iron phosphate batteries, they'll take larger materials and grind them down. We can make incredibly small particles, 2, 3, 4 nanometers in size by growing them up. And so we look at the ability to do that. We look at the ability to add additives, mix new materials together. So most of it has been based on, we haven't invented a new material for a battery material. We've invented new synthesis materials, new sizes, new ways of putting them together, nanowire materials, new architectures. But we have, I guess I was going to show this tomorrow, we have made materials that haven't never been made before. We made this material, aluminum oxynitride, which is a transparent bulletproof material. It's from Star Trek, I guess. I've never seen Star Trek. I asked my students to see if they could make transparent armor. And then a couple weeks later, they came back with aluminum oxynitride-based materials, so not completely transparent yet. But in that case, it's the first time it's ever been made as an aerogel, so we made aerogels of that material. So we're materials scientists. We look at what materials are currently being made, what ones are possible, what are their limitations. People have asked me why I don't have a battery company. I have natural gas, and I have electronics and environmental remediation and medicine. I think battery companies are really hard. And what we look at is our strategy in that area is new form factors. And our strategy is structural batteries for UAV applications and such.
Okay. There's another question.
I was just wondering, you're working with Phage Display, and I suppose you have a resolution of around 1 in 10 to the 9 or something. Would you agree?
For the number of individual phage?
Yeah, the library is around 10 to the 9.
That's what they say.
Yeah.
Yes, that's what they say. It's not true.
Would you be helped to get a 1,000-fold increase in the library?
Yeah, and so
because then you could go into in vitro library.
Yeah.
But then you couldn't do it with phage.
Right.
You had to do it, but you could find the candidate and then put it back into the phage.
Yeah, so maybe we can talk more about that offline. So that's a question we get quite a bit of. First of all, are you taking full advantage of your 10 to the 9th library? And we're not, actually, because when we go in and we do an individual panning, we're only actually screening a small part of that 10 to the 9th to begin with. And then, truth be told, when you go away from the P3 protein, which is the one that's involved in infection, and you go to the major coat protein, you drop to about 10 to the 6th in the ability. And that's not based on complexity of DNA. It's based on sequences that you can actually express, which is actually very different. And so I think that having a larger library that you can actually address in a single pan, all billion, would be a huge advantage. Right now, I forget what my students have shown over and over again, and it's against what people say in the literature, is that you're usually only seeing a small part of that. And part of that is not based on DNA sequence. Part of it is based on expression and populations that take over as you do your screening.
Okay, are there any further questions? So if not, I think we are going to meet Angela tomorrow again. And before leaving the stage, I have to give you something as tradition from, I'm not giving you the SciFinder bag, but I'm going to give you something that we have tradition here with the Hassel lecture, is giving the Hassel lecturer a book that has been actually edited in a very limited number. There are only 100 issues of this.
Wow.
And this is number 47.
Oh,
that
thank you.
corresponds to 2017.
Yes.
And that is actually in German.
Okay. I'll use Google Translate. Yeah. Thank you so much.
Thank you very much for a very great lecture.
Thank
and
you.
please join me again for thanking Angela.